I'm going to turn to one of my colleagues, Yana Najjar, who is in Chicago today for an academic meeting that she had, an industrial academic meeting she has, and turn the podium to her virtually. Yana, take it away.
Thank you, John. Good morning, everybody. As Dr. Kirkwood mentioned, I'm out of town for a meeting, so I'm sorry I couldn't be there in person. I'm one of the melanoma medical oncologists here at the UPMC Hillman Cancer Center, and I thought it would be very germane to start off by talking about clinical trials. You all hear us talking about them all day, every day, and I think we often take for granted that what we mean is widely understood, and so I was really hoping to go over that today. So throughout history, we know, of course, cancer is not a new problem. People have been dealing with cancer since prehistory, no doubt, but people have been living longer and longer, and the longer we live, the more likely we are to develop malignancy. And our methods of dealing with this initially were quite rudimentary. With surgery, we used to inject bacteria into the tumors, hoping to elicit an immune response. Believe it or not, within the last 120 years, we then became a bit more sophisticated with radiation, with chemotherapy, and then a very, very rapid domino effect, and I mean that in the best way possible, with the development of multiple new therapies spearheaded by immunotherapy in melanoma. What I want to really highlight to you, and this is one of the great privileges of being a melanoma oncologist, of all of the approvals that I've put here for you, showing you the pace at which things have happened. The first for most things in the modern era was in melanoma. The first cytokine therapy, that was high-dose IL-2, eventually approved by the FDAs in the 90s for melanoma. High-dose interferon, developed, of course, by Dr. Kirkwood. Immunotherapies, be it first generation with ipilimumab, second generation nivolumab and pembrolizumab. The first oncolytic virotherapy, TVEC. The first combinations of immunotherapy, ipi-nivo, rela-nivo, the first bispecific antibody, tebentafusp, and the first cell therapy, as Dr. Kirkwood alluded to, lifileucel. Every one of these firsts was in melanoma, and that's something I think about all the time, and I think is really quite remarkable. As the history of cancer therapy was evolving, that really moves in tandem with the history of clinical trials. The first experiments to really resemble clinical trials happened 500 BC, but more towards the modern era, the first randomized trial, and when we say randomization, we mean somebody is getting drug A, somebody is getting drug B, but the decision of what to do is random, meaning which patient gets on which arm of therapy is random, and that's really to minimize bias. That happened during World War II for the treatment of tuberculosis. And even if you were to look back to the mid-1700s, the first trials of an intervention was for treating scurvy on a boat. And so clinical trials are certainly not new, but I wanted to touch today on the modern era. There are a lot of different treatments available for melanoma. We have the wonderful privilege at this point, and most of us trained in the era before we had any of this. When I was a fellow now almost 10 years ago, we were just starting to use immunotherapy, and we were seeing the results of those clinical trials. But as you can see here, we have a lot of different options, and a lot of these options have arisen in the several in the few years prior. So this has really led to a complete paradigm shift in how patients do. The overall survival for a patient with stage 4 melanoma used to be in the order of months. It was, frankly, always a terrible discussion. And we now find ourselves with 10-year survival data showing us that almost half of patients are alive and cancer-free at 10 years. That is remarkable. Needless to say, there's a lot of work to do. We all show up to work every single day trying to figure out how we can extend the benefits of immunotherapy to the 50% of patients who are not benefiting. But that is not the topic of our talk today. Today, I really wanted to focus on clinical trials. And what I want to drive home is, if nothing else, is that we got to the point where we have trials, where we have treatments, and where patients are actually surviving because of clinical trials. every treatment that every single patient has received was tested at some point or is currently being tested in a clinical trial, and that is without a single exception. And so I hope I've convinced you that clinical trials are important, because it is only through clinical research that we gain insights and answers about how safe and how effective treatments may be. And these advances that have led to significantly improved overall survival that was frankly unimaginable a decade ago, is only because of participation of patients in the context of talking about malignancy. And the discoveries arising from these trials have saved innumerable lives. So, what is a clinical trial? Quite simply, it is a way to look at new ways to either detect, prevent, or treat disease. And clinical trials can ask different questions, and they can also have different focuses. But largely speaking, in the context of cancer, in clinical trials, we study either new drugs, new combinations of drugs, or new ways of using currently existing therapies. And the goal of clinical trial is to determine if a particular approach is safe and effective in a certain population. Why would people take part in a clinical trial? Well, of course, there are many different reasons. One is altruism. People take part, and this is always, as a physician, really humbling to see. People take part. In my experience, more often than not, they say, well, even if it doesn't help me, it might help science. It might help move the field forward. So altruism, of course, is a big driver. There's also the chance, depending on the kind of trial, to have access to a treatment to which we otherwise have no access to. So, for example, today you can get ipilimumab and nivolumab, but 10 years ago, the only way you could get ipilimumab and nivolumab is if you enrolled in a trial and you happened to be in the 1/3 of that clinical trial that got the combination. In some cases, there's also potentially added care, added attention from the clinical trial team. And what I mean by that is, if you were to get the clinical trial treatment, you would be observed more frequently for side effects. You may or may not have more frequent blood tests. You may or may not have more frequent scans and physician visits. So we have been involved in a lot of different trials at the UPMC Hillman Cancer Center. And in the very top of the slide, I am showing you the drugs that are currently FDA approved for metastatic disease. And believe it or not, we have been involved in every one of them that I can think of. And the cycle of science is such that once we find that a drug is effective in stage 4 disease, we ask ourselves, might it be effective to prevent melanoma coming back in stage 3 and stage 2 disease? And guess what? We've been involved in all of those as well. And that's really a huge privilege of working where we all work. And I'm just showing you what's FDA approved so far. There is a lot more to come. Myself and all of my partners, Dr. Kirkwood, Dr. Devar, Dr. Luke, Dr. Zarur, we are all involved in a lot of different clinical trials that I hope will extend the timeline you're seeing here with new therapies added every single year. Where does a research idea come from? Usually, it comes from one of two places. It's either the lab or it's from an observation that we make in the clinic. And once we have an idea, and when I say we, I mean a physician scientist such as myself or any one of my colleagues, all of whom do the same thing, or a scientist in the lab, a basic scientist, somebody who studies biology or immunology. Once we have an idea, we test this idea in the lab and in animals. And this is what we call preclinical data. This is setting the stage for taking a specific drug or combination of drugs or old drug in a new way into the clinic. And the treatments then move through a series of steps, what we call phases of clinical trial. What I want to highlight to you is that it really and truly takes a village. So before any trial gets to any patient, there has been a ton of work at the back end. And when I think about how we conceive of clinical trials, I've highlighted here in red researchers that can often be a physician or a PhD scientist working in very close collaboration with lots of different stakeholders, statisticians, bioinformaticians, other scientists and the study sponsor, that's ultimately, if the study moves forward, the person who, I shouldn't say the person, the organization or the foundation that funds the science, the scientific work, but is not actually performing the studies themselves. So you can see here in the life cycle, if you will, of a clinical study, there's the research hypothesis and the development of the protocol. This has all happened before it comes to you at all. The second circle is subject enrollment. And what I really want to highlight to you is that the enrollment of the patient, of course, is the whole point. It's why we do all of this. But a clinical trial is a very living, dynamic, I think of it as a very breathing thing that is always changing. And there is always input from so many different team members as we continue to learn from the data that we are collecting, as we analyze the data, as we manage the trial, and as we move towards, if appropriate, submission to regulatory authorities of the data for FDA approval. There are many different phases of clinical trials, and here at the UPMC Hillman Cancer Center, we're involved in all of them. A phase 1 trial is what is often thought of as a first-in-human trial. And what I mean by that is that there's a fair bit of science, really a huge bit of science, that has led to us thinking that drug X might be of interest. But the first question, of course, is this safe to give to a human being? And so the overarching goal of a phase 1 clinical trial is to evaluate the safety. That's the take home. Once you establish that a drug is safe, you turn your attention a little bit towards the effectiveness of the drug. A phase 2 trial is really to see if there's a good, robust signal of efficacy of a drug in a certain disease state. For example, immunotherapy X in melanoma. In addition to testing the efficacy of the drug, we further evaluate safety. If we are enthused by the efficacy data that we see, we then move to phase 3. And the goal of a phase 3 trial, and this is really a behemoth of an undertaking with many hundreds of patients, if not thousands in certain disease states enrolled, is to confirm the effectiveness of the intervention compared to, phase 3 trials will invariably have a comparator, compared to what we have now to see if it's as good or better, or and we continue to monitor the side effects throughout. And it's a phase 3 trial more often than not, although nothing is black and white in medicine, but more often than not, a phase 3 trial is what will lead you to regulatory approval, although there have certainly been drugs approved on the basis of phase 2. A clinical trial follows a plan known as a protocol. And a protocol is really the document, if you will, that outlines what we're doing. And we write our clinical protocols and we have this very thoughtful design so that we're balancing the potential benefits versus the risks to participants. And I always teach our trainees, and we have all been taught that in a clinical trial, more often than not, usually gets one really good question. You cannot ask if a drug is better than the other drugs and it's safe, and you cannot ask if it's better in 10 different ways. This is all very regimented as it ought to be. And so I think of it as you get one main question, and that's what we call the primary objective. So for example, the goal of the clinical trial is to test if drug A in patients with advanced melanoma increases the period of time before the cancer gets worse, or increases survival. You generally will pick one main question and build the whole statistics of the trial. For example, how many patients do you need to enroll around that main question, what we call the primary objective. And in the protocol, we put all the details. If somebody were to come from outer space and to read the protocol, it should make sense. And the questions that we highlight there are who is eligible, who is not eligible, how are we protecting participants, what are we doing? What are the tests? How often? What's the treatment? What's the dose? How do we modify the dose if we need to? How do we monitor for toxicities? How long does treatment go? And what data are we gathering? Trials are led by principal investigators in the context of melanoma, physicians, and members of the study team continue to monitor patients' progress and safety. Now, the informed consent process, of course, is at the heart of all of this because we want to make sure that you understand what you're going into. And I have had many patients say, I'll just sign. And we say, no, that's not the way to do it. I really need you to read over this. Let's go through it together. Let's make sure you understand what is being asked of you here. And so we go through the informed consent document, outlining the details of the study, everything I've already referred to. And I want to highlight that that process continues. So if we learn something additional about the safety of a drug while you're on the trial, we have an updated informed consent. I'm sure many of you have signed these many, many times. Once you have gone through all of this, you then decide whether to sign the document. And of course, clinical trial enrollment is voluntary, and you can withdraw your consent at any time, no ifs, ands, or buts. From a patient perspective, once you consent, once you say, okay, I want to proceed, we then as a team go through what's called a screening process. We make sure that we have, I think of it as all our ducks in a row. We make sure that you as an individual meet the eligibility criteria, the inclusion criteria. You have no exclusion criteria, up-to-date scans, do we need biopsies? You then go into treatment and surveillance. So what should you ask if you're offered a clinical trial? I would say to understand the purpose of the study itself is probably the chief consideration. And then, of course, all the nuts and bolts. What treatment is being administered? How is your safety being monitored? How are the results being monitored? How long will it last? What is expected of you as a participant if you take part? What do we know about the toxicity profile so far, if anything? Sometimes in phase 1, we don't know very much, and we discuss all of that with you. Other things that I think are important to discuss are what are the alternatives as well. Cost, of course, is a big one, and we have a whole fiscal office that helps us figure that out. What will be billed to your insurance versus what does the study pay for? And also to know who's your point person to ask questions to. I always tell my patients when they're coming to discuss a clinical trial, the more ears, the better. Bring whoever you can with you. I have no doubt that it's very overwhelming. Think ahead of time about what you want to ask. Don't hesitate to write it down. Also, don't hesitate to jump in with new questions as they arise. I personally never mind if patients ask if they can record what I'm saying. I don't think your physician will mind if you ask them, so that you can go back to it for more time to absorb all the information. And of course, it's okay to take time to think about it. In fact, more often than not, we say take the informed consent document and let's regroup next week. Needless to say, we're not cowboys. It's not the Wild West. These trials are not happening in a vacuum. There is very strict oversight. That could be the topic of a whole day discussion of how we got to the level of oversight we have and why. There are strict ethical guidelines, both to protect you, the participant, and to protect and preserve the integrity of the science. The IRB, or the Institutional Review Board, is an independent committee. It's made up of physicians, patient advocates, community members, statisticians, other physicians, to really ensure that the clinical trial is ethical and that your rights are protected. After a clinical trial is completed, we then carefully examine all of the information that we have collected, and we decide what to then do with it. For example, if it was a phase trial, is it safe? Do we want to move to phase 2? If it was phase 2, does it seem to be effective? Have we confirmed it's safe? Shall we move to phase 3? We then publish our findings in peer-reviewed journals. Peer-reviewed means that other scientists are looking at the data and asking robust questions, and we make sure that this is an unbiased process in which we all share in the scientific enterprise. And once a new approach has been proven to be safe and effective in a clinical trial, it may then become a new standard of care in medical practice, which is literally what has happened with every single one of these drugs that I outline here, which many of you have been treated with in the past. This is a really important one, and I wanted to take a moment to address how does the doctor decide what trial to put a patient on? And I tell every patient that while we have reason to believe that a treatment may be effective, and certainly we can discuss why we think treatment X may be effective, we absolutely do not know if it will be effective. You can do the entire trial and enroll 500 patients in a phase 3 trial and find that the new drug is no better than what you already had. That absolutely happens. But if you don't do the trial, you don't really ask the question, you don't know. So as a clinical investigator, equipoise is key. And what I mean by equipoise is going into a trial, enrolling a patient, really not knowing which is which is the right answer, if you will. We go into it understanding that we don't know, and that your clinical situation as a patient warrants enrollment in this clinical trial, that we think this clinical trial may benefit you. It makes sense to enroll, but of course we don't know. We consider a lot of information for every single patient. What's the preclinical data? Why are we enthused about this approach? The patient's own beliefs and value system, regardless of the way I feel about it. I vaccinate my own children. There are people in the world who are not into vaccine, and so a clinical trial vaccine, a clinical trial looking at a vaccine is not right for that patient. If I have a patient who's a Jehovah's Witness and they're not going to want blood or platelet transfusions, then certain clinical trials where we know we're going to need those are not appropriate. So we look at the patient themselves, of course. We look at the phase of the trial and where the patient is in their own disease journey. We also look at the anticipated side effect profile, if we think the patient will be able to withstand it, and also whether the individual patient meets the inclusion and exclusion criteria. And I want to really stress, and I hope you all know, and Dr. Kirkwood touched on this, we are all here for you. We are here in this with you. And it would be very obnoxious to say we understand what you're going through as patients and as caregivers. We absolutely don't. But we are here every day trying to move the needle, trying to find more effective therapies as we continue to move towards a cure. And thank you so much to every one of you for all that you do in terms of volunteering your bodies, your time, your efforts, your resources to help us to advance science. And with that, I'll be happy to try and answer any questions that I can.